This dissection was done in order to study mammalian anatomy of a fetal pig 1 to 2 weeks away from birth. Red latex solution was injected into the arterial system and appears red to show arteries, and blue latex solution was injected to the venous system to make veins appear blue. The purpose is to gain an understanding of how organs work together and to gain a better understanding of the mammalian anatomy and its distinct characteristics. This is a comparison of the anatomy of a fetal pig and the anatomy of a human. Hypothesis. If the pig is a mammal, then it will exhibit similar organ function structure to that of a human. Methods. Tools used include a dissection kit including a scalpel, scissors, pipettes, paper towels, pins, rubber gloves, styrofoam trays, cardboard trays, aprons, goggles, and string. The sample was a female fetal pig 1 to 2 weeks away from birth. The first step of the procedure was to place a fetal pig in an anatomical position and secure its limbs to the tray using pins and string. We made a midline cut running from the top of the thoracic cavity to the end of the abdominal cavity. At the diaphragm, we cut anteriorly up to the throat region. We made a lateral cut at the left and right side of the midline incision, made a lateral cut on each side of the midline, posterior to the forelimbs and anterior to the diaphragm to expose the thoracic cavity. We cut posteriorly from the diaphragm to the umbilical cord, leaving a flap containing the umbilical cord by cutting around it. We made cuts on each side of the midline incision, posterior to the diaphragm, to expose the abdominal cavity. Once both cavities were open, we proceeded in cleaning the pig in order to locate and remove all of the organs. Their hearts, like humans, have 4 chambers: the right atrium, right ventricle, left atrium, and left ventricle. Food moves through their digestive system from the esophagus into the stomach, through the duodenum, into the small intestine, then into the large intestine before it is excreted. Here you can see where the diaphragm separates the thoracic cavity from the abdominal cavity, with the diaphragm lying directly beneath the heart and above the liver. With the membranes connecting the small and large intestines cut and the intestines being laid out, they measure 2.3 meters in length. The liver is made up of several lobes and performs many functions for the body. Here you can see the spleen attached to the stomach. Here you can see the kidneys, which are located beneath the other organs in the abdominal cavity. The heart and lungs. The thyroid gland, thymus gland, larynx, pharynx, esophagus, and trachea are all found in the throat region. The major parts of the reproductive system consist of the ovaries, fallopian tubes, and uterus, which is not shown. The umbilical cord, which supplies nutrients, oxygen, and blood from the mother to the fetus. The thyroid and thymus gland, which produce essential hormones. The kidneys. After removing the organs, we examined the anatomy of each. Then we proceeded to dissect the brain by removing the fetal pig's head and making incisions from the top of the eyes to the base of the neck to remove the skin and muscle covering the skull. We used a scalpel to cut along the sutures of the fetal pig's skull. Then we chipped away at the skull cap with the scalpel, exposing the brain. We cut the spinal cord and any connecting tissue to lift the brain from the skull. We then examined the brain and compared it to a human brain. Here are the olfactory bulbs, which help with the sense of smell. Using a scalpel, we separated the brain into right and left hemispheres. Here you can see the different lobes and regions of the brain. Both the pig and human brain are enclosed by the skull and have nervous tissue surrounding it. The average weight of a pig brain is 180 grams, while the average weight of a human brain ranges from 1,300 to 1,400 grams. Our conclusion. Our hypothesis was correct. The pig had similar characteristics, organ structure, and function to that of a human. Like humans, there were 4 chambers of the heart in the pig and the same vessels and brain structure. All organs were similar in placement when comparing them in a human.